Good evening. My name is Antti Vuolanto. I'm the CEO of Herantis Pharma. So Herantis, our ambition is to be the first company developing disease-modifying therapy for Parkinson's disease, so to stop the progression of the disease. Herantis is a public company listed here in Finland, in Helsinki First North. So the necessary forward-looking statements and then going into the actual topic. So our lead asset, called HER-096, is a small peptide that is derived from a neurotrophic factor called CDNF, or cerebral dopamine neurotrophic factor. With HER-096, based on the data that we have from preclinical settings, we can protect the dopamine neurons from further degeneration, and we can also support their functional restoration. With the peptide HER-096, we can also have very efficient brain penetration. So the program is currently just finalized phase 1. So we had 2 phase 1 studies, phase 1A and phase 1B, where we have established very solid safety data. We have shown very efficient brain penetration. We have been able to select a dose for phase 2. We are still waiting for the biomarker data from the Parkinson's patients from the phase 1B to be reported by mid-January next year. And on a high level, the summary for the next steps would be to find a development partner and/or financing to conduct a phase 2 efficacy trial in Parkinson's disease. So Parkinson's disease is a disease of degeneration of dopaminergic neurons responsible of controlling movements. The underlying reason for the degeneration and eventual cell death is not known. However, a lot of related to the pathology on the neuronal level is actually known. However, the current treatments which exist, they can treat the symptoms, so they can deliver more dopamine into the region in one way or the other, and in a way affect the symptoms. However, they cannot affect the disease progression at all. So basically, when a patient gets the diagnosis, they already at that stage know that, okay, my symptoms can be treated for some period of time. It can be longer, it can be shorter, but there will be a period of time when the quality of life of the patients will start deteriorating, and there is nothing that the current treatments can do for that. And that is, of course, something that we want to change at Herantis Pharma. So there is a high unmet clinical need, and of course it means that there is also a huge market. So it has been estimated by Parkinson's News Today in the springtime that the management of Parkinson's disease, the annual cost globally is over $250 billion. The drug market size currently is between $5 and $6 billion. However, this is estimated to double to $13 billion by 2033. Currently, there are 10 to 12 million patients. It has been estimated that the number will double until the year 2050. So we have a huge unmet clinical need. There is huge market, and we definitely want to be, if not the first one, at least among the first ones to address this. So how we do this? As mentioned, the lead compound HER-096 is based on CDNF. And CDNF was actually the starting point of the company over 15 years ago, so we have a very long experience with the same biology. So we have some key evidence from CDNF that really makes us enthusiastic about the opportunities in Parkinson's disease. Now, what you see on this slide is data from a preclinical study with non-human primates who had induced Parkinson's-like pathology. And you see 2 like images having some staining, and these represent staining images from these animals and from the region where these neurons are located. On the left-hand side, the placebo one, there, of course, the animals have been treated with placebo infusions for 3 months, and on the right hand with CDNF. And you don't need to be an expert seeing that with the CDNF treated, you see much more intense dark coloring, and the dark coloring represents the dopaminergic neurons and their connections to other neurons. So by naked eye, you can see that yes, we have been able to induce neurorestoration with CDNF that actually coincided with improved motor symptoms and non-motor symptoms. Based on these great results, we did also a clinical study with intracranial administration of CDNF. And among these 17 fairly advanced Parkinson's patients, we actually were able to show several individual subjects that showed signals of clinical improvements or biological responses. For example, in the graph that represents one patient who was first 6 months on the placebo, then 6 months on the active CDNF treatment, and then in follow-up period. The solid line represents the motor scores, the clinical assessment of the motor functions, and during the placebo period, it was rather stable. Switching to active CDNF treatment, we see a decline, which means improvement, then there is another line, dashed line, which is the standard of care treatment dose. And as you can see, at the same time, when the CDNF kicked in and we see an improvement, also the medication had to be, in a way, lowered. And this is, of course, an indication that the natural dopamine production by the neurons had been, in a way, increased by CDNF administration. Of course, great results, but the challenge was the intracranial administration. And that's why we developed HER96. That has exactly the same mechanism of action, but we can use subcutaneous administration. So basically, HER96 fully captures the activity. It mimics the active site or the binding site of CDNF protein into its pathway that CDNF controls naturally, which is called unfolded protein response pathway. So CDNF is a natural protein in humans and is known to promote the neuronal cell survival and functional recovery, and that's its normal biological function. So with HER96, we can do the same. So we can reduce the cell stress, we can reduce protein misfolding, and we can reduce neuroinflammation. So we can protect the dopamine neurons from degeneration and support their functional restoration. I mentioned that no one knows what triggers the pathology. However, we know a lot what is going on the neuronal level. And the degeneration of the neurons is driven by a vicious cycle that is composed of accumulation of toxic alpha-synuclein aggregates, cell stress or ER stress, and neuroinflammation, which is also driven by ER stress. And all of this results in the degeneration of the dopaminergic neuron, resulting in decreased dopamine secretion and movement disorder. And when this vicious cycle continues, eventually the dopaminergic neuron dies. However, with HER96 administration, we can break down this vicious cycle. And we are actually hitting two objects at the same time. So we are hitting the unfolded protein response pathway simultaneously in the neurons, but also in the glial cells responsible of the neuroinflammation. So we can normalize a very basic pathway, the target pathway, unfolded protein response pathway, and thereby normalize the function of the dopaminergic neuron and increase the dopamine secretion. And all of this we have shown in preclinical settings that yes, HER96 really captures this activity. And this is nicely shown in the video, which many of you have maybe seen already earlier. So this is a bar walking test for motor coordination. The first one was just a normal aged mouse. The second one has the induced pathology, and the pathology has exactly these different features that I showed, the vicious cycle going on, and it results in a very clear motor phenotype, so struggling with the motor coordination. And then the third mouse here has the induced pathology, but has been treated with HER96. And yes, as you can see very clearly, we have been really able to affect the symptoms, so we have been able to break down the vicious cycle and improve the symptoms and improve or increase the amount of dopamine in the brain region. With HER96, as mentioned, we have completed phase 1 program. We had 2 studies. First, we did a phase 1A clinical study in healthy volunteers. It was a single ascending dose study with 6 different dose levels, and here we established safety in healthy volunteers. The pharmacokinetics, so the behavior of the compound in human body, and also we demonstrated efficient brain penetration. Then we conducted a phase 1B clinical trial, where for the first time HER96 was administered into Parkinson's patients. The treatment was for 4 weeks, 2 subcutaneous administrations per week. Here again, we showed very nice safety profile. We further studied the pharmacokinetics. We also established very efficient brain penetration in Parkinson's patients. And we were able to, based on these data, to select the dosing for phase 2. And as already mentioned, we are currently still waiting for the biomarker data to be completed. And there, actually, what the objective is to demonstrate the biological response to HER96 dosing. So preferably in biomarkers on individual or pathway level, in such a way that we know that it's linked to mechanism of action and also linked to the findings in preclinical settings. Then we are at the same time, we are planning for phase 2 efficacy trial. We expect the phase 2 to be started by the end of the next year. Of course, still pending for partner or financing for that. But then we also want to start exploring HER96 in other indications, as the basic biology is very common with other neurodegeneration or aging-related indications. And related to the resourcing of phase 2, we are really deeply or intimately looking at partnering. We have obtained a lot of interest from the pharma based on these phase 1B data, and of course, the coming biomarker data will be also key for this. And we are going to JP Morgan meeting in early January to continue these discussions. We are looking at different potential partnering models. So far we are in a way open and let's see how it goes. But we are also at the same time looking at potential equity financing and non-dilutive financing. On that side, good to remind you that we already have a good level of external validation of HER96 and the biology. So the phase 1B was financed by a consortium of Michael J. Fox Foundation, Parkinson's UK. But in addition to that, we also have an investment commitment from EIC funds, so they can invest equity into the company. They still have almost 12 million to be deployed to Herantis. So basically, this was, in a way, the summary of Herantis, and I really want to emphasize that we believe that HER96 is a potentially game-changing therapy that really can become the first disease-modifying and neurorestorative treatment for Parkinson's disease. All right, thank you. And then I guess we'll continue with Q&A.
Thank you, Antti, and welcome to the Q&A session.
Thanks.
You already mentioned a little bit about the biomarker data release and analysis, but maybe it's worth it to go through it again. So what are you extracting from that data when it comes out? What is the meaning for you?
Yes. So in preclinical settings, we of course have very thoroughly analyzed what is going on a tissue level and cellular level in the animal models, and we know what kind of changes we see as what would be in a way the starting point for those changes that will then result eventually into, let's say, clinical or symptomatic or disease modification improvements. And now in clinical samples, we want to show that yes, we have started exactly the same changes as what we have seen in preclinical settings, and what are in a way based on our modeling, those ones that we should see in patients as well. And if we can do that, it of course decreases the risk of failure, because we can show that yes, we have initiated a cascade that hopefully and potentially will eventually result in clinical improvement of the patients.
So it's not efficacy signals, but it's kind of signals of the drug doing what you would like it to do.
Exactly. That's the meaning. Yes. We need to remember that the phase 1b clinical trial was only 1 month treatment, and for a chronic disease like Parkinson's disease, which is slowly progressing, we should not expect to see an efficacy signal within such a short period of time. But showing that we have, in a way, started the biological process, that will be a strong signal for partnering.
Great. Then you recently reported on a 6-month preclinical toxicology study. So can you recap what was done and what were the results?
Yeah, so we have earlier, of course, completed a 28-day tox study. That was a requirement for starting the phase 1 program. Going towards phase 2, when we expect to have either 6 or 12 months treatment of Parkinson's patients, we of course need to have a longer toxicology program in preclinical settings. And this is exactly the 6-month study. So we had 1 animal species, rats, where we studied 6 months dosing, and of course the objective is to show that it is safe and well tolerated. And this is what we were able to show, that the results were very well aligned with the 28-day tox data. So everything is cleared for starting a clinical trial in efficacy trial, 6 months or 12 months dosing with HER96.
Okay, that's good. That's good. Okay. Then about the upcoming phase 2 trial. So you don't have the exact plans announced yet on it, but as I understand, the duration of the study and follow-up time of the patients will be relatively short, maybe 6 months, like in your animal trial, or 12 months or something like that. But the Parkinson's disease takes a little bit or a long time to develop. So how do you balance this kind of relatively short-term trial to the challenge of showing efficacy, which would benefit from a longer trial? So what's the trade-off and balance here?
That is exactly the very good question that I think Herantis and the whole industry is currently, in a way, considering how to balance between the selection of the patients, the duration of the study, the number of patients, and then the primary endpoint and other endpoint assessments. And there, I think, there are a couple of key things. How to select the right patients so that we have as homogeneous patient population in the treatment arm and placebo arm as possible, and how we characterize the baseline characteristics so that we can actually have the possibility to see a change. And as you said, rightly so, that 6 months or 12 months, they are still relatively short period of time for Parkinson's disease. So we need to have a measurable change expected during that time. And there, the right endpoints comes into play. Traditionally, it has been a clinical endpoint in all the symptomatic trials. So it will be like neurologist assessment at the clinical observation how the motor functions are developing. However, the industry is currently moving towards having digital assessments, so either a smartphone-based or a wearable device that monitors the symptoms either passively or then by using active tasks. And this is, of course, something that we are currently looking at, what would be the best outcome measure so that we can optimize both the patient, the size of the patient population and the duration of the study. And there, of course, we need to also understand what is the biomarker signal and what is the time frame of when we see the biomarker signal in the phase 1b data, so that we can predict how long study we should have in phase 2 in order to see a clinical benefit in in that study.
Do you think the biomarkers can serve as an endpoint, or is it more for the partnering discussions?
Biomarkers, for us, it will be an important tool for designing the phase 2, exactly the kinetics. It will also be a risk mitigation factor for partners and possible also for investors. We are not expecting to have a very fixed biomarker endpoints yet in phase 2, the next phase 2 trial, but it is possible that we have a hypothesis that we will test in phase 2 so that we can use them later on, for example, in pivotal trials.
And the digital tools you mentioned, are they the kind of tools that are already being used, or have they been used in other trials?
Yes, there are many platforms that have been used in Parkinson's trials and in some other indications as well. There is currently one phase 2 clinical trial in the US that has a primary endpoint based on active smartphone-based assessment of motor symptoms. The company is called Neuron 23. But there are basically all the studies that are currently involving disease-modifying therapies. They have a digital measurement platform somewhere in their endpoints. So this is definitely something that is widely used, and I expect that, let's say, within the next 5 to 10 years, we will also see digital endpoints as regulatory endpoints in either Parkinson's disease or other neurodegenerative diseases.
Great. And you already talked a little bit about the funding for phase 2, but maybe we can go through that again. So you have ongoing partnering discussions, and the biomarker data will also be a part of that. So how do you see the timeline and the position you are in for those negotiations?
I think our position in those discussions is really strong. If you look at the competition currently, there are maybe a handful of compounds in phase 2 or going into phase 3 development. So actually the pipeline is pretty dry. However, the unmet clinical need is high, and the market opportunity is huge. So we have got really high interest on phase 1b data and of course the previous data as well. But of course the time will tell whether the phase 1b data alone is enough to trigger a partnering agreement, or if the partnering agreement terms that might be available for us are sufficiently well that it justifies going forward with those. And that's why we are also looking at equity financing at the same time. So we are not in a way in a situation that we don't have any options. We want to have now the options open. And of course during this spring, Q1, Q2, that's the time of finding the let's say the practical possibilities, and then decide what is the best way going forward. And on the partnering side, as I have mentioned in many previous presentations as well, we are looking both at the global partnering, but also more regional partnering. That could also be a good way of financing phase 2 program.
Yes, and I guess the phase 1 results are kind of very solid in those terms for undertaking the discussions. it seems to me that since last summer or during this autumn, the financing activity, at least in the US, has picked up, and there's more M&As, more licensing deals. Do you see that kind of uplift in Europe and in your area?
Absolutely, we see that. So I think the improvement in the biotech financing has started. It started with bigger deals. If you look at what is happening in US currently, there are like financing deals between 100 and 500 million US dollars. Not so many IPOs yet in US, so they are being like established companies having like on market utilities. However, my assumption and based on my discussions with several US banks is that IPO activity will start picking up, and also the number of deals will be increasing during the next spring, and it has already started. So I think there will be a keen interest to find good opportunities. And moreover, there have been a couple of very big M&A deals during the autumn overall in drug development, and it means that there are certain investors who have quite a bit of liquid money to be deployed to other companies as well.
Okay, so let's take the final question. You mentioned the pipeline for Parkinson's being quite dry, but are there some positive or interesting examples that you would lift from Parkinson's research or other neurodegenerative research that is kind of good or interesting signals? Something that has happened during this year, or maybe some readouts you are expecting next year?
Yeah, I think it's a good signal that Roche is going forward with their immunotherapy against alpha-synuclein aggregates. It means really that big pharma is interested in heavily invest in Parkinson's disease development. And we see that only as a positive signal. We don't, in a way, see that as that we would be in a competition with Roche. I think if they can prove to be efficacious and they can be they prove that they can continue efficient development in phase 2, it is also very beneficial for Herantis. And based on, in a way, mechanistic thinking also, it might be a good opportunity also to combine Herantis, HER96 and such immunotherapy then going forward when these new disease-modifying therapies are on the market.
Thank you very much for the discussion and presentation, and good luck for the next year.
Thank you.
All right, dear viewers, thank you very much for joining tonight's Inderes Life Science Night, and looking forward to talking to you again in the next events, and please read our latest analysis and comments on inderes.fi and .com.